Ecological station, which is about an hour from MSU's main campus. And I study all the soil bacteria and fungi that live underground and do some amazing things that we're going to tell you about today.
Hi, everybody. My name's Allison Bender. I'm the outreach coordinator for the Great Lakes Bioenergy Research Center, and it's my job to help share the science that happens in our center with people like you.
And I'm Adriana Trusiak, and I'm a research coordinator at the Great Lakes Bioenergy Research Center, and I help to coordinate all the research being done. And I get to work with a great group of scientists like we have here. So now to start, I'm gonna share the video highlighting the work we've done on a research project to study root interactions in soils. We hope you enjoy. And then we're going to have a Q&A after with everyone.
Soil supports all terrestrial life on our planet. You might not think about soil often, but it is the source of most of our food. The health of our soil and understanding how it functions in relationship to the plants and microbes in its environment is critical to me, to you, and to everyone on Earth. Soils can also benefit the climate. As we continue to use fossil fuels to make electricity, heat our homes and buildings, and power our travel, we've been increasing the amount of heat-trapping greenhouse gases in our atmosphere. This results in more variable and extreme weather. While the scale of climate change is massive, we have many solutions that can help us reverse track. Soil might play a key role. Soil can counteract the carbon dioxide emissions from fossil fuels by absorbing carbon dioxide from the atmosphere and storing it deep in the ground. It is estimated that soils store 3 times as much carbon as all the plants on Earth. Some soils can store more carbon than others. Increasing soil carbon, or how much carbon is stored in soil, is a win-win solution. We can get more productive soils for agriculture and decrease the amount of carbon dioxide in the atmosphere that causes climate change. We now know that carbon in the soil is really good, but how does it actually get there? Most of it comes from plants. Plants undergo a process called photosynthesis, where they take in carbon dioxide from the atmosphere and use it to build their shoots and roots. The more carbon plants put underground or into the soil, the better. We can grow plants that both store carbon in the soil and can be used to produce fuel for boats and planes that are currently powered with fossil fuels. The Great Lakes Bioenergy Research Center studies how certain plants can be grown and harvested to produce sustainable, renewable fuel that is good for the climate and for the land where the plants are grown. The above-ground part of the plant is used to make fuel, while the plant's roots grow deep into the ground and build soil carbon.
Soil can achieve an increase in the soil carbon storage when the amount of the carbon added to the soil exceeds the amount that is being decomposed and returned to the atmosphere as a carbon dioxide. Therefore, there are 2 fundamental ways to achieve soil carbon gains: by adding more new carbon into the soil, or by maximizing stabilization and the protection of the soil carbon, both the newly incorporated one and that already present in the soil.
Some crops, especially perennial grasses like switchgrass, poplar trees, and mixed native grasses, are especially good at building soil carbon for reasons that we don't fully understand. Partly it's because we don't till the soil every year when we plant these perennial crops. And when we till the soil, we tend to stimulate the microbes that then produce CO2 from stored carbon. And partly it's because the roots are in the soil year-round instead of just for a few weeks, as is the case for annual crops.
Great Lakes Bioenergy Research Center scientists want to determine how to increase the amount of soil carbon in the ground.
Different bioenergy crops have the different carbon storage capability. We were thinking, why is that? Who makes this difference? So my research questions came from here. So we can use the 2 contrasting bioenergy crop fields, one with only one species, switchgrass. Another one is restored prairie with 18 different plant species, including the switchgrass. So we are going to look a little closer into this field, and it can be trials to see each plant and soil under the plant.
If plants can interact underground, that makes us wonder, could neighboring plants' interactions impact soil carbon?
Above ground, we see diverse plantings of different plant species, but we're just now getting the techniques to be able to study this below ground. And one thing that I've learned is that depending on who a plant neighbors, it interacts with different microbes. And one of the ways it does this is by the carbon that it puts underground. So plant roots secrete carbon-rich sugar substrates called root exudates. And these root exudates are really critical for plants' ability to communicate with both these different plant neighbors and with the microbes that it's interacting with. A previous study I've done showed that the composition of these root exudates, so maybe the different sugars or the different acids that a plant is putting out through its roots, changes depending on who these neighbors are. And so if those root exudates change depending on who a neighbor is, there's a high chance that that's also affecting the amount of carbon below ground.
Roots and the exudates they leak impact the soil's carbon storage because the carbon is passing directly from the air, through the plants, and into the soil. Another pathway for carbon to get from the atmosphere through the plants and into the soil is through fungi. Plants indirectly exchange carbon from one neighbor to another through fungal hyphae that connect them underground.
Plants evolve with fungi as helpers, as friends. And so there's many groups of fungi that depend on plants, and plants depend on them for proper nutrition. These are called mycorrhizal fungi, and they are kind of an extension of the plant root system. So they help the plants obtain water, they help the plants obtain phosphorus, and they also help absorb nitrogen and transfer this to the plants. And so we think about them as biofertilizers. Their filaments are like 1/100th the diameter of my hair. So they're very, very small. And this allows them to transgress across and within soil pores. And so they're able to mine out nutrients where plant roots would not have access to them.
Once carbon is in the soils, it's stored in soil pores. The structure of the pores impacts carbon storage.
Basically, soil pore functions as a channels of the air, water, and the nutrient in the soil environment, but they also can be the habitats for the microorganism in the soil, which use the soil carbon as their energy source. That carbon which leave microbes' body as a non-gaseous form. That would be the carbon that can be most effectively protected from the further decomposition. Such carbon compounds can be easily tied to the surface of the soil, like clay particles, or they can also move into the very small soil pores. And in there, carbon can be protected for a long time, like hundreds or even thousands of years.
Carbon storage in soils also depends on tiny soil microorganisms like bacteria. They use carbon as their energy source. While some of the carbon will be lost to the atmosphere as carbon dioxide, a part of it will stay remaining in the soil.
In 1 teaspoon of soil, you'll have billions of bacterial cells. So there are like many of this tiny microbes in the soil. What bacteria does is it uses the complex organic compounds that are available from the plant roots in the form of like root exudates or the rhizodeposits, those kind of stuff. So it takes those complex organic compounds and break down them into simpler form and uses them as an energy sources. And the simpler form is then gives it back to the plant. So it's like an exchange trade between the microbe and the plant where they help each other.
To break down carbon in soils, microbes need enzymes.
Microbes are very small and they can't eat their carbon foods and digest them inside of their body because these carbon molecules are too big for them. So they secrete these enzymes to the soil and let these carbon compounds cut into smaller pieces in the soil around them and then absorb those cut down pieces. This is why the enzymes we find in the soil are called extracellular enzymes. If you want to see how active the soil microbes are, looking at the extracellular enzyme activity is a good choice because it tells us how hard they're trying to eat their foods and what kind of food they're targeting to eat, and most importantly, how much carbon they can potentially eat.
As you can see, there are lots of different things that might influence how soil carbon accumulates. We designed an experiment to get to the nitty-gritty of how
plant interactions,
root exudates,
fungi and bacteria,
soil pores,
Enzymes
all impact carbon storage in soils.
How do we address all these research questions and see the roots without disturbing the plants while they're growing?
So we started designing the rhizoboxes late 2019. We went through a couple iterations before we settled on this like 3-compartment design. We actually had to build all these from the ground up. So we purchased like a ton of plastic and just used a bunch of cement, a lot of bolts and screws, and assembled these. And they're like half a meter long and like a third of a meter high. So they're quite big. Each box needed like a few kilos of soil. To fill them, what we had to do is we wanted to pack them to a certain bulk density and a certain moisture level. And just because of how they're set up, of how thin and tall they are, we couldn't bring them to the certain moisture level without adding the moisture as we were filling. It took us like 7 days, and we had like 14 different people come through, and everybody was packing boxes, just in layers, adding water, put another layer of soil, and then after we reached the top, then we compressed them all, put the bolts on, and moved to the next box.
The rhizoboxes will help us cross multiple scales to understand how tiny things like soil pores and microbes ultimately can have huge impacts on plant communities and global carbon storage. How can we tell what carbon came into the soil from plants? And how can we tell from which plant the carbon came?
You can track the carbon by using isotopic elements. So we use 13C. It's a stable isotope. You find it in nature naturally. So our boxes have 3 plants. We make it so the plants we don't want to uptake this 13C, we cover them. So then they're not photosynthesizing. Think about it just like going to bed, right? You're not going to be eating. You're not going to be walking around the house. And that's exactly what these plants are doing when they're covered. They're just kind of, they're waiting to get some sunlight so they can become active again, you know? So the plant that we allow to take up that 13C takes it up through its leaves, and then it distributes it all across the plant and wherever it's growing. It also uses it in exudates on the roots. And then these microbes live on these roots, and they'll eat up those exudates, and they'll reproduce, and they'll grow these communities. And then when you have like roots that interact with each other, these different neighbors, then you can get the communities that kind of like live on both roots. They kind of transfer the carbon that way. So it's really interesting that you can start to see your 13C carbon in plants that you didn't allow to uptake it through the air. It's actually transferred through the roots.
So we grew these plants at an angle, and that allowed the roots to grow on the surface of this rhizobox. And when doing that, we could visualize where the roots were. So we carefully used sterilized tweezers and little spatulas to dig the root, almost like an archaeologist. We were excavating these roots out of the soil to carefully collect them. So after excavating this root, we put them in a plastic tube, and we fill the plastic tube with glass beads. And the role of this glass beads is to stimulate a soil environment where the root is interacting with different pores and different sizes of soil particles, but it's glass, and so it's sterile. And that allows us to extract all of these carbon-rich compounds or root exudates that they're pushing out.
After 8 months of preparation is over, the most exciting and important part, sampling the plants and soil, begins.
For my study, I specialize in looking at the microbes in their habitat. That is like how they're located in the environment they're living. So I look at microbes living in soil, on the soil matrix, or on the plant roots. What we do is like we take like a gram of soil, and then there are different methods. There are like general stains that can be used where you can stain the cell wall of a bacteria and look at, you can quantify them. Or there are some advanced techniques such as FISH, which is fluorescent in situ hybridization. So it's like a probe which goes and binds to the RNA of the microbe, which has, it's like a small sequence of the complementary RNA, So if you know that small sequence, you can like add an additional dye of your choice. So when it goes and binds to the RNA, it fluoresces, like it glows, and then you can visualize it under the microscope so that we can use it in undisturbed soil samples. And the good thing is like we can use different type of dyes. So just imagine like a blank canvas and you have like all different type of bacteria. And for each particular bacterial group, you have different kind of dyes. So it will be a wonderful, colorful picture with all different type of bacteria showing like how they live. And bacteria is usually in form of small colonies. But with this technique, with this fish technique, we can see individual cells, how they are like grouped into and where they are situated in their habitat.
First, I take small rhizoboxes from our rhizoboxes, and I will map the soil enzyme activities on the soil surface. But since these enzymes are just liquid in the soil, we can't see them. So we can use these artificial carbon compounds that becomes fluorescent when they are cut by these enzymes. I will attach a membrane soaked with this carbon compound and put it on the soil surface. Then the enzymes in the soil will react with the compounds and become fluorescent. In this way, we can map the enzyme activity by capturing this fluorescence. And then these fluorescents are only visible when they are with these ultraviolet lights. Once we have that, just normal DSLR cameras can capture those fluorescences. There are many ways to analyze this. For example, if we want to find the hotspots, which means like very high activity area, we just like set the threshold values and then only select the soil area where the enzyme activities are like above certain value. Also, we can separate out the root areas and then just look at the enzyme activity on those roots.
The researchers are currently analyzing results that will then be written up and published. However, the work, curiosity, and questioning won't stop there.
So if these root exudates and if these different plant-neighbor interactions are in fact changing these soil microbial communities, does that have effect on future plant growth, or does that affect how these plants are interacting? So in 1 year, switchgrass might be growing with certain neighbors, and the next year, maybe a different neighbor comes in, and do the microbes that it grew with in the first year affect how it interacts with that plant in the second year? This is something that's really hard to study in the field because you can't control who those neighbors are or who's in the soil. But these plant soil feedback experiments allow us to more easily get at these questions.
With more data analysis and experiments on the way, our researchers are working to better understand how interactions with the community of plants and microbes around bioenergy crops can help them store even more carbon in the soil. As we learn more about the potential to grow plants for fuel, we can work with farmers to grow crops that take up more carbon from the atmosphere and store it in the soil for longer periods of time. The ongoing collaborative research at the Great Lakes Bioenergy Research Center is just one of the ways in which we can all work together to protect our soils and to reduce the threats of climate change. To learn more about our work and to get involved, visit glbrc.org.
So welcome back, everyone. Hope you enjoyed the video. So now feel free to send any questions in Q&A, or if you're watching through Facebook Live, post them in comments. And I believe Hazel is monitoring that, so maybe she can feed us the questions. So there is one question: do the extracellular enzymes come from living organisms?
Thank you for the question. Yes, mostly. So extracellular enzymes are some kind of like saliva for us, right? So yeah, it's coming from the living organisms. But since soil has so many organisms that's already died, some of the extracellular enzyme is just remaining in there. So yeah, half is coming from the living ones, but maybe another half would come from already dead ones. That would be the answer.
Great. Thanks, Alyssa. Another question is, how might drought conditions affect overall carbon storage in soils?
I think I can answer for that. Actually, it really depends on the soil condition, but soil properties and the condition. But drought condition can make the microbes less effective, and maybe the carbon storage created by the microbes can be lower than the other just regular condition if the soil is too dry.
Great. Anyone wants to add anything or?
I think that's great, JinHo. I think one other thing I might add in is that we're finding that the things like these root exudates, the different compositions of the different types of chemicals and compounds that these plants are putting out can change a lot under drought. And we're still learning about how those changes might affect the amount of carbon that's stored, but it definitely changes those microbial communities and so has a potential to influence those dynamics. And especially with increasing droughts, there's a lot of research going on at Michigan State University to learn more about how droughts are affecting these below ground processes. That's a great question.
We have another really good question. So corn is currently grown for bioenergy. Is that a good plant to use, or doesn't it store enough carbon in the soils? Anyone want to take that?
I could start. It is a good plant to use in terms of renewable energy if you build like ethanol out of it. But the problem is that in terms of just like soil, it's not great. It takes a lot out of the soil and doesn't add a lot back to it. So we're exploring all these different plants with the GLBRC, the Great Lakes Bioenergy Research Center, that look at plants that we can use for renewable energy that also don't negatively impact the soil.
I think one key thing that's really special about these other crops that are different from corn is that corn is an annual, which means it needs to be planted every single year. And so you can imagine that that takes a lot of fossil fuel just to plant and manage that corn every single year. But for instance, the switchgrass that we use in this study and prairie and some other crops are perennials, which means they don't have to be planted every year. And that would mean a lot less fossil fuels would be grown to actually then create the energy. So you would end up with net higher energy when you subtract all of the energy that's used for production, which is something that's worth considering that we're thinking about here at the Great Lakes Bioenergy Research Center.
And one more point to add on to that is the Great Lakes Bioenergy Research Center makes a point to not use crops that are used for food to kind of avoid this food and fuel debate. So that's another reason for the crops that we do choose to study.
To add one more thing, so compared to the corn, I think switchgrass is a native prairie species living in North America. So it not only stores carbon, we can keep like biodiversity in the ecosystem. So we can expect many like related ecosystem functions from switchgrass as well.
Yeah, these are all great points. And just to emphasize, yeah, really not trying to compete with the food crops is a really big thing. And also like following on Alyssa's point, is not only that it's native species, but also a lot of these species can be grown on lands that have been abandoned. So maybe agricultural lands that are no longer productive and cannot support food production, can support production of bioenergy crops. So we have another question. Do the roots themselves add carbon to the soils when they die? And how much carbon comes from dead roots versus root exudates?
You take a stab at this one. This is a great question, and JinHo's looking at the roots too, so I'll let him jump in. Yes, a lot of carbon comes from those roots when they're dying because those microbes are eating them and breaking that carbon down, and so those roots end up being carbon. And a lot's in the exudates too. Whether we get more carbon from dead roots versus root exudates, I don't think we know that. And I think one of the reasons we don't know that is because it's really hard to study these root exudates in the field. So we have things like greenhouse studies where we can look at them, but whether those rates are really comparable to what's happening in for instance, a 6-year-old switchgrass plant is really difficult to know. I would think that there could be more coming from root exudates because it happens more continuously and it's coming from all these different roots, but I might be biased because that's what I study. So I don't know. It's a great question, and I think something that scientists are really thinking about in the coming years. JinHo, do you have something to add?
Yeah, I think you answered that really nicely, but I also add something more. Actually, the fundamental concept of the carbon storage is the imbalance between the input and the output of the carbon source. And input can be the root and the root's exudate, and output is the decomposition from the soil, decomposition of the root and the root exudate. And yeah, it's really depending on the soil condition, like the pore structure as we showed, and soil moisture someone asked to us. But yeah, so that's the reason why we are doing the research on that. And yeah, we should look that in overall, considering the pores and the roots exudate and the decomposed mass of the root, something like that.
Can I add something? So I personally look at the root decomposition for my entire thesis. And then, Taylor's right, we don't know the answer, but carbon use efficiency of the microbes is really important. And there are some studies that's talking about the root decomposition, and when they are processed by microbes, the carbon tends to be more stabilized. That means the carbon can be stored in the soil longer time. So yeah, we still don't know the answer, but many researchers are working on that, I can say.
Great, thank you all. I don't see more questions on chat, but I have a question to all of you. This was a very extensive experiment that took many long hours and a lot of collaboration. What was your favorite part of it? What did you enjoy the most?
Oh, the silence. Definitely for me, it was like the actual collaboration. We worked with, there's like 3 completely different labs that put a lot of man hours into it. And I got to meet a lot of people who I wouldn't have normally had the opportunity to work with. Great researchers, great people, connections and friends I'll have for a while.
I think I would echo that. This was one of the larger collaborations I've done in my whole PhD, the largest. And I think when you get a lot of different people thinking about a topic from different angles, there's a lot of synergy and it's really exciting. And I learned things about my specific questions that I wouldn't have thought about if I wasn't in this group. And it was also really exciting. There was a lot of prep work, just thinking about how do we build these boxes? How do we sample them? We all want to sample different things. And so there was just a lot of time, and then coming together on the first day. First day was rough and it took a long time because we had a lot of different pieces to work together. But by the end, we each had our role and it was kind of like clockwork. And that was really amazing to see that come together.
Yeah, this collaboration broadened my perspectives on the carbon storage. Actually, I learned a lot of things about the microbes from Archana and the Lutz exudates from the Tayler and the extracellular enzyme from the Ayssa. So I had no idea about that before I started this experiment, but now I got a little bit more than before.
I will also talk about the friendship and partnership. It was COVID situation, so we were working at home, but this collaboration made us meet personally and work together like all day long. So that was the happy moment.
Great, that's so good to hear that. And thank you all again for all your work you put into it, and we're very excited to see the results. I want to thank all the attendees for your great questions and for attending and learning more about bioenergy. The video will be available at the MSU Science Festival Facebook page, and it will be on our YouTube for you to watch later. So thank you all again.
Yeah, I just want to echo that. Thank you to all of our presenters and attendees. This was awesome. Thank you all.
Thank you.
Thank you.
Thank you. Thank you so much.
Thanks. Have a great night.
Bye.